Hello, welcome back to my channel. So we are going to learn on subtopic 2.2 for today, biology Kssm form 5. Okay, the main organ for gases exchange in plants. Okay, right. So I hope you are ready with your pen and paper and do take notes and let's do this. Okay, as usual, before we go on into the lesson, we are going to see the learning standards. Okay, so here you must be able to justify the necessity of gases exchange in plants. Okay, and then explain the mechanism of stomatal opening and closing. Okay, so there are two factors here. Okay, the uptake of potassium ions and also the changes in sucrose concentration. So what happens with this that causes the stoma to open or close? All right, and then to conduct an experiment, okay, to compare the stomatal distribution on upper and lower epidermis, okay, on monocotyledon and eudicotyledon leaves. So this one we won't do it, but I'll explain a little bit, okay, on what you can expect from the experiment. Okay, and lastly is to predict with explanation. Okay, predict, but you must be able to explain also like why, okay, on the effect of water deficiency on plants on stomatal opening and also closing. So if there's lack of water, what happens to the stoma? All right, so basically that's all for the learning standard. Okay, now the necessity of gases exchange in plants. Now, unlike animals that search for their own food, we can, if humans, we can just go and open the fridge and find our own food. Okay, but plants, on the other hand, they can't walk to the fridge. I mean, yeah, but okay, they can synthesize their own food through photosynthesis. Okay, so in order to carry out photosynthesis efficiently, plants need to exchange gases and also to absorb light. Okay, so gases exchange in plants enables plants to obtain, what? Carbon dioxide for photosynthesis. Okay, and also carbon dioxide from the atmosphere will diffuse through the stomata into the leaf during the day when the light intensity is high. Okay, and carbon dioxide is absorbed by the palisade mesophyll cells. You have seen in the previous subtopic, okay, subtopic 2.1, the palisade mesophyll and also the spongy mesophyll, okay, which has a lot of chloroplasts. Okay, so this carbon dioxide is also absorbed because carbon dioxide is used in photosynthesis, okay, to form glucose, which is the product of photosynthesis. Okay. Okay, now the exchange of oxygen and carbon dioxide between the plants and the surrounding occurs through the stomata. Okay, but it also occurs through a structure called lenticels, okay, which are found on the stems. Okay, let's look into the detail into it. Okay, stomatas are the pores that are located on the lower epidermis of the leaf. Okay, and each stoma is guarded by a pair of guard cells. If you can look at the stoma, okay, stoma is actually an opening, just a hole there. Okay, and it is guarded by the guard cells. Okay, so we have the guard cells here. Okay, and this is the stoma. All right, this is the stoma, which is surrounded by epidermal cells. Okay, so each stoma, which is guarded by the guard cells, okay, they will control the opening and the closing of the stoma, okay, by changing their shapes. So later we will see how it changes its shapes. Okay, and now the guard cells contain chloroplast also, okay, to carry out photosynthesis. Okay, lenticels on the other hand, okay, it allows diffusion of gases in and out of the stems and also the roots because the structure is found there. Okay, but it's not as complex as guard cells and stoma. So gases exchange occurs, okay, through diffusion, through stomata and also lenticels. Okay, now let's look at the mechanism of stomatal opening and closing in general. Okay, so we must know that oxygen and carbon dioxide diffuse in and out of the leaf through the stomata. Okay, the mechanism of stomatal opening and closing depends on the conditions of guard cells. Okay, whether they are turgid or whether they are flaccid. Okay, turgid and flaccid, you know these terms from chapter 3, form 4 Kssm. Okay, so mechanism of stomatal opening, and depends on whether the gut cells is turgid or flaccid. Okay, so the condition of the gut cells, whether it is turgid or flaccid, depends on two things. Okay, the potassium uptake, okay, by the potassium uptake by the cells, or the sucrose concentration in the gut cells. Okay, uptake of potassium in gut cells, and also sucrose concentration in the gut cells. Okay, so if you look at the sucrose concentration, okay, during the daytime, okay, or in the presence of light, photosynthesis will take place. Okay, and it will produce sugar or sucrose. All right. So during the night time, when there is no light, sugar will be converted into starch and stored in the plant. All right. And then here we have potassium ion uptake. Okay, potassium ion which will accumulated or eliminated. So this will determine whether the gut cells becomes turgid or becomes flaccid. Okay, so when this happens, when there is more potassium ion or when there is less potassium ion in the gut cells, the solute potential will change. Water potential will either increase or decrease. Okay, water will either diffuse in or diffuse out from the gut cells through osmosis, and the gut cells becomes either turgid or flaccid. So this is in general, but the flow is the same. Okay, later you will see the slides whether for stomata opening and also for stomata closing, you will see the two difference, but the flow is the same. All right, now let's look at the slide, the next slide. Okay, now let's look at the opening of stomata. All right, now the mechanism of stomata opening, okay, it involves photosynthesis. Okay, when photosynthesis happen, there is a high concentration of sucrose in the gut cells. Okay, so during the day or in the presence of light, photosynthesis takes place in the gut cells. Okay, sucrose is formed and the concentration of sucrose increases in the gut cells. Okay, sucrose concentration increases, and then the water potential, the osmotic pressure first will increase. Okay, so when that happens, the sucrose concentration increases. Okay, water potential will decrease. So what happened? The water will diffuse into the gut cells through osmosis, and then the gut cells become turgid and curve outwards, so the stoma opens. This is for sucrose concentration. But what about the uptake of potassium ion by gut cells? Now, potassium ions, okay, are pumped, okay, pumped into the gut cells. Okay, so this will actually increases the solute potential in the gut cells. Okay, and water potential in the gut cells now will decrease as well. So water potential decrease, water will diffuse into the gut cells through osmosis from high concentration, okay, in the epidermal cells into the low concentration in the gut cells. So water diffuse in. So when the water diffuse in, gut cells will become turgid, curve outwards, and stoma open. So you see this part? Okay, this part here is the same. Okay, water diffuse in, cell become turgid and curve outwards, stoma open. So basically these two factors involved in the opening of stoma. Same thing goes in the closing. Now let's look at the next slide for closing. Okay, now during the closing of stoma happens when there is a low concentration of sucrose in the gut cells. Okay, at night or in the absence of photosynthesis, okay, where photosynthesis does not take place. So what happened is that the sucrose concentration in the gut cells, okay, what happened? Decreases. Sucrose concentration decreases. Now the water potential will increase. That means there's a lot of water in the gut cell. So water will need to diffuse out by osmosis. So water go out, diffuse out by osmosis. So when water diffuse out, the gut cells has less water, so the cell become flaccid. So become flaccid, it curves inwards and the stoma closes. Same goes with the uptake of potassium. Now, when there is no photosynthesis, there is no need of potassium, so potassium ion is eliminated by the gut cells. So the solute potential, the potassium concentration decreases. Solute potential decreases. Again, water potential increases. So in the gut cells, a lot of water, water diffuse out from the gut cells into the epidermal cells by osmosis. Gut cells become flaccid, curve inwards, stoma closes. So basically, same concept. Too much of sucrose, too much of potassium in the gut cells, water potential decreases, so water diffuse in cell becomes turgid and stoma open. But when there is no photosynthesis, sucrose concentration, potassium ion concentration decreases. Okay, when that decreases, water potential will increase, so water move out. Okay, by osmosis, diffuse out by osmosis, cells becomes flaccid. Okay, when they become flaccid, they will curve inwards. When they curve inwards, stoma closes. Is it clear? I hope it's clear. Okay, if no, you can just let me know in the description box below. In the comment box below. Okay, so yeah, this is one of an example, okay, of cactus where they open their stoma only during the night for carbon dioxide because of the temperature, too high temperature, because they live in a desert, right? So colder temperature at night can reduce the loss of water. So yeah, that is one difference in plants at different region. Okay, so this is the experiment that I told you about, okay, on counting the number of stomata, okay, found on the monocot and the eudicot leaf. Okay, so whether there are more, stoma at the upper epidermis or more stoma at the lower epidermis. Okay, so in eudicot leaves, okay, there are actually more stomata on the lower epidermis than the upper epidermis. So low have more stomata compared to the upper epidermis in eudicot leaf. But in monocot, okay, the number of stomata on the upper epidermis and also lower epidermis is almost the same. Okay, so this is a different, that maybe a difference in one or two or maybe less, but it's almost the same number. But in eudicot, you can see the number totally different. Okay, upper epidermis have lesser stomata compared to the lower epidermis. Okay, so there's a video here by Cikgu Fazli. Okay, you can I will put this in the description box below. Okay, so you can go and actually have a look. Okay, have a look at this video, and he will show you on how to conduct this experiment. Okay. Okay, now the effect of water deficiency in plants on stomatal opening and also closing. Okay, so here we know that water from plant is lost in the form of water vapor to the surroundings through stomata. Okay, now when stomata opens widely, the rate of water loss from the plant is high. Okay, so the opening and the closing of the stoma is dependent on the turgor pressure of the gut cells, whether the pressure is high or the pressure is low. So the gut cells will either be turgid or will be flaccid. Okay, so during drought, now what happens during drought is that the plants will experience water deficiency. Correct. So when it has less water, the plant has to respond very quickly to reduce water loss. Okay, by transpiration. Okay, so water loss is through transpiration. So how to reduce the transpiration rate? Of course, close the stoma. When you close the stoma, water loss is reduced. Okay, so stomata closure is the first reaction to the drought stress experienced by most plants. So by closing the stomata, less water is lost. The plant will actually increase its efficiency to use the water. Okay, so and this will also actually prevent carbon dioxide from diffusing into the leaf, hence reducing photosynthesis rate. So the plant is really in stress, in drought. Okay, that's what happens. Okay, so it helps the plant to survive through the drought and do not wilt easily. Okay. Okay, now let's revise a little bit. Okay, let's see if you remember what we have discussed. Now, true or false? Gut cells that are turgid causes stoma to close. Turgid, open. Okay, so false. When the cells are turgid, the stoma opens. When the cells are flaccid, the stoma closes. Gut cells. Okay, now second one, plant carry out gaseous exchange through lenticels and stoma. Yes, of course. Alright, number 3. Gaseous exchange in leaves occurs through lenticels. In leaf. In leaf, it occurs through stomata. Okay, so lenticels is found in the stems and the roots, so it's false. Now, during daytime, number 4, at high light intensity, stoma open. Yes, because it needs to carry out photosynthesis. It needs the water, it needs the carbon dioxide, and at high light intensity, a lot of sunlight, perfect, photosynthesis on. Okay, and number 5, during drought, plants respond quickly by closing the stomata. Yes, of course. So that is true. You can't see, but yeah, it's true. Okay, so it has to close the stomata to reduce water loss. Okay, with that we are done. So let's do formative practice 2.2. As usual, formative practice comes after the lesson. Okay, so now question number 1, state the difference of gases exchange in plants during respiration and also photosynthesis. Differences. So differences, the keyword is whereas. In photosynthesis, what happens? Whereas in respiration, what happens? So this is the keyword. You must use this. Okay, and number 2, how does humidity in the surrounding affect the size of stomatal opening? So high humidity, what happens? Stoma open or closes, and low humidity, stoma closes or open. Okay, number 3, stomatal distribution on the lower leaf surface is more packed as compared to the upper leaf surface. Justify. Why? Why do we need more stomata below the epidermis and not at above the epidermis? Okay, so relate with the function of the stoma. And what is the importance of stomatal closure when the plant lacks of water? Why is it important? Okay, so think about it, do it, and then you can check the answers in the description box below. Okay. So with that, we are done. Okay, we are done, and that's all for today. Thank you for watching, and please don't forget to like and subscribe to my channel. Don't forget, please. Okay, that's all. Bye bye.